And now we'll go for the next talk, which will be by Victor Greiff. So Victor is Associate Professor for the Systems Immunology at the University of Oslo, and he's going to introduce his work on computational and machine learning approaches for identifying disease or antigen-specific information in adaptive immune receptor repertoires. So please, Victor.
Thanks so much, Enkanita, for the nice introduction, and thanks so much for inviting me to this meeting. Yes, so my talk will be about computational approaches to immune receptor analysis. And before I go on, I would like to preface that this talk or this work that I'm presenting here is in very close collaboration with another group at the University of Oslo. More information can be found here. Most of the work that we are performing on adaptive immune receptor data relies on the main premise that adaptive immunity is a flash drive that records immune events. So, for example, if we encounter immune events such as bacteria, virus, or vaccine, B cells and T cells engage with these agents and clonally expand, build memory, and so on and so forth. And we are using high-throughput sequencing. We are able to gather all this information by sequencing DNA or RNA of these B cell and T cells. Which are, and we are especially interested in the B cell receptor and the T cell receptor, or antibody when it's in soluble form. And I think what we are mainly interested in, most of us that are working in this field, is connecting the information of the data that we get from these immune receptors to immune status, so health or disease, or antigen binding. And both of these sides of adaptive immune receptor analysis I will show in this talk. So within each individual, we have a huge diversity of T cell receptors and B cell receptors, and the potential diversity out of which this is sampled is huge and kind of still unknown. And what I just said is that, okay, so we assume that each immune event that we encounter, so for example, vaccination, infection, or autoimmunity, is to some degree recorded in the adaptive immune repertoire, such that we can, by screening those receptors, these large data sets, perform diagnostics just by looking at the B cell receptor and T cell receptor data sets, in order to then maybe even in the future prognostically predict health or disease or different disease statuses. And on the sequence level, one of the main questions in the field is, can we predict binding from the antibody or T cell receptor towards the antigen or even the epitope? Yes, and both of these things I will show a bit in this talk. So now we are on the repertoire level, where where my group is quite interested in is to what extent is actually immune status impacting the immune repertoire. So how similar are immune repertoires across individuals, let's say across health, but also health versus disease. And to that end, we have developed a framework that looks at many different dimensions or features of the immune repertoire, such that overlap, positional amino acid frequencies, subsequence occurrence, which are also called k-mers, clonal diversity, germline gene usage, and sequence similarity. And based on these features, we have built a similarity score, which I'm not going into detail here because of the brevity of my time here. But so based on these features, we can build a similarity score which allows us then to compare repertoires all versus all. And if we do that on a very large data set of more than 1,000 repertoires of health, RA and SLE, so health versus autoimmunity, we see that overall, the similarity score that ranges between 0 and 1, 1 being very similar, 0 being not similar at all, with respect to all these repertoire features that I just showed you, we don't see on this global level, if we just look at unsorted T-cell receptors from the blood, we don't see a difference between those immune states. And which one could see as a potential problem for what we want to do, which is immune receptor diagnostics. But I will briefly, I will show you on the next slide why this is not such a huge problem. So we did this also on a very large data set of CMV infected individuals, so CMV positive and CMV negative, which was published by Emerson et al. In 2017. And here we again find no difference between CMV positive and CMV negative individuals on this global level, which is also in line with the findings by Emerson et al., which found that by applying very sophisticated statistical approaches, they could only find 164 clones that actually could differentiate between CMV positive and negative individuals. So showing that only a very small fraction of the repertoire is probably differing in the blood in these immune statuses between health or disease, or between case and control. So which poses a problem for machine learning, or for trying to differentiate between health and disease, could pose a problem. So that's why we need machine learning to find those really small patterns, really lowly concentrated patterns in the repertoire, in order to tell us these different immune statuses. And the machine learning case that we are after for this question is the repertoire classification case, where we have a repertoire of, for example, 2 different immune states. And we want to find those sequences that predict health versus disease. And the problem with applying any kind of machine learning to immune receptor research is that the biology of adaptive immune receptor repertoire poses a lot of problems to machine learning, which is that there is quite low clonal overlap across individuals. So one has to kind of constantly make sure that one is not comparing apples to oranges. What we would like, of course, to do in the future is not only to classify health versus CMV, or CMV positive versus CMV negative, but also classify many different diseases, right? So multi-class and multi-label approaches. Then we know that there are different factors that can influence the immune receptor repertoire. We know from especially autoimmune diseases that the signal can be quite low in the repertoire. We know that there is polyreactivity, which can further increase the challenges for machine learning. We know that the signal is distributed in the frequency space, in the sequence space, and in the structural space. Then there are confounding factors, such as, for example, age or MHC. And then there are all these technological factors that also other people, I think, will talk in this session more about how we in the future must be able to standardize these factors in order to take them, in order to be able to remove them from the machine learning analysis. Nevertheless, and there are many people that have already tried this task of immune receptor machine learning to classifying immune state. And some of these people are also in this session. nevertheless, one of the main problems, in addition to the problems that I just outlined, is that on the machine learning side, most of these approaches interdependently have not been very flexible to reuse for other approaches. The reproducibility is not really there yet, and the reusability especially. And the structure of air data has been rarely exploited for machine learning. So that's why we developed this machine learning ecosystem for immune receptor analysis, which is called ImmunoML, which addresses most of these current technical challenges. You can use it for repertoire-based classification, sequence-based classification, and also for benchmarking new machine learning approaches that you are currently developing. And it reads in most of the data out there in a very standardized fashion. You can perform most of the machine learning approaches that you find out there, and if not, you can implement your own and add it. And it is reproducible, and importantly, I think it also has a immunologist-friendly interface, which takes away most of the machine learning lingo and lets also immunologists try machine learning on immune receptor data. Importantly, I would like to also mention that it integrates with the iReceptor database, which is a large database that assembles in a very standardized fashion immune receptor data. There are many billion sequences in this database, and you can basically stream from this database into ImmunoML your data and then perform your machine learning analysis. What we are currently working on now, so despite there being large data sets out there, these are not really sufficient to benchmark in a very fine-grained fashion machine learning approaches for airr-c data. So that's why we are developing. So we have previously developed a simulation framework for immune receptor analysis, which is called ImmuneSim. And now we are updating it to further increase its ability to really implant small and very sophisticated immune signals into the simulated data to really represent native data sets. So this is ongoing work. And but nevertheless, we have already applied these simulated data to a benchmarking study that we just recently put out there on bioRxiv, where we trained 1,300 machine learning models on 850 data sets, so on 210,000 immune receptor repertoires, so something that you really cannot do with experimental data. And we basically, and with this analysis, you can now see how changing parameters in your data. So for example, the amount of signal that we have in the data, which is called the witness rate, and here the sample size, so the number of repertoires, how this scales with the prediction accuracy that we then get. And here it was a binary case, health versus disease, such that you can then outline which conditions can already be predicted well by very baseline machine learning approaches, which was the research question here. So for example, we found that with a very simple machine learning approach, we can get very high prediction accuracy when the immune signal only occurs in 1 out of 50,000 TCRs. So even quite simple machine learning approaches perform already quite well on very difficult question, which is encouraging. So as a summary of the first section, we found that on non-sorted immune repertoires, the signal is quite low. So probably in the future, one needs to really look for cell population specific signals. And of course, in infection or acute infections, the signal is much higher. But in very diffuse diseases or autoimmune disease, for example, it will be quite difficult to not look at very specific cell populations to find a defined signal that one can use for diagnosis. Then we developed this ImmunoML machine learning ecosystem, which integrates with simulation frameworks and also benchmarking frameworks. So this benchmarking study that I showed last was performed entirely in this ImmunoML framework. In the last 5 minutes, I will focus on something else, so which is on integrating structural data with sequence data, which I think people have just started to incorporate into one another. So, and here the question is, we are now on the sequence level and trying to see, can we predict antibody binding based on the antibody sequence and the antigen sequence? And ideally, even better, where, so if they bind, where on the antigen would it bind? So paratope epitope prediction. And this task is very difficult because basically binding is governed by a very small region of the antibody, the complementarity determining region 3. And there is this large polyreactive space that we have to take into account, and these large non-linear dependencies imposed by the structural folding of the antibody, especially in the CDR3 loops. And this is important for antibody discovery. So human antibody discovery, as you know, is very slow, and nature is very, very fast. Once we get infected, within days we have quite good antibodies for most diseases. And how can we learn from this? So we have to learn from nature. And we have to perform, again, large-scale simulations to improve our machine learning approaches for antibody-antigen binding prediction. So, and until now, the question, or until recently, the question of predictability of antibody-antigen binding was a bit unanswered, I would say. And the state of the art was that people thought that epitopes are mostly indistinguishable from other parts of the protein surface if we don't have the antibody. And we knew that paratopes have some kind of enriched, some sort of enrichment of amino acids. But nobody had really looked at, or very few people, groups had looked at those paratope epitope pairs together in a combined data analysis. And that's what we did. And we kind of hypothesized that if we can find motifs that are shared across different antibody-antigen structures, then paratope epitope prediction might be possible. And this is what we showed in a recently published paper. I will not go into further detail. This is the title of the paper, where we showed that there's a set of paratope epitope motifs that enables predictability of antibody-antigen binding. Unfortunately, we show predictability, but the predictive performance is still quite low, since what is available out there are only 1,000 non-redundant antibody-antigen crystal structures, which is far not enough for really learning the underlying rules of antibody-antigen binding. So what we are now developing is a antibody-antigen simulation framework, where we can simulate antibody-antigen binding at very large scale in 3D. So we get, for each simulated crystal structure, paratope, epitope, and affinity. And we were now able, with this framework, to simulate data sets that are surpassing an orders of magnitude what we have available experimentally. So we have now 1 billion antibody-antigen crystal structures with affinity. And we also show in this paper that we will hopefully put out soon as a preprint, that our simulation framework recapitulates the biological complexity of antibody-antigen binding quite well. And we are now able to really benchmark machine learning approaches and find what data do we need in the future in order to really predict antibody-antigen binding experimentally also. And we find, again, with this framework, that if we have structural information, it really helps prediction. So sequence to sequence prediction is still very, very difficult, even with very large data sets that we have here. And we also use this framework to show that we can perform antibody design at an unconstrained scale using machine learning. So here we use generative learning, where we put the simulated sequences into a generative framework and then test whether the generated sequences again bind the antigen using our simulation framework. And we find that generative machine learning approaches can simulate or can generate large numbers of sequences that are again antigen specific. So this is kind of a proof of principle that we can indeed use generative machine learning for antibody design and discovery. Yes. And with that, I am coming to the end of this talk. So I showed you on the second part of this talk that antigen binding is predictable. What we need now is large-scale data, and the best thing that we have so far in terms of large scale is synthetic data, which we can use as ground truth to really develop new machine learning approaches that we can then use on the experimental data once these become available. And what we are now doing on the immune status prediction side is exploring causal analysis in order to account for confounding factors, and also further develop these machine learning approaches and perform large-scale benchmarking studies. And on the antibody antigen side, we are looking at in vitro validating our simulation framework and also the generative framework, and also perform more benchmarking efforts. Yes. With that, I would like to thank all the collaborators at the University of Oslo and across the globe, our funders. And I would like to highlight again that the airr community has many different outlets where you can learn more about the state-of-the-art research that is currently going on in the airr field. Especially, I think the YouTube and the webinar are very useful also for beginners in this field. And we have a Slack for answering any kind of experimental and computational questions. And on Twitter, we tweet the most current papers in the field. So, and with that, thank you very much for your attention.
Thank you, Victor. So waiting for eventually some question. Actually, I have one question. Well, I have several questions, but so,
I kind of know, yeah.
Regarding your machine learning approach, so you said that machine learning, and we know it's hard because there are many parameters that we should take into account. One parameter that people usually, at least in the TCR field, raise is the importance of the HLA. So how do you deal with this? Are you studying mainly repertoires from HLA matched donors, or do you think that we can eventually look at more broadly to identify some patterns?
Yeah, so I think there are several answers to this question. So I think there are some papers, also Emerson, I think, have shown, and you can correct me if I'm wrong, but that their predictor worked across HLA types. So sometimes you can develop predictors that are valid across HLA, which is of course great. But I agree, and that I mentioned on the last slide, that we need to learn more about how to address these confounding factors. So how do we actually simulate? So, and I think we do not know enough about how strong is the signal of HLA on the repertoire and on prediction accuracy. I think we need more data on this, and we need to understand better the biological structure in order to better simulate it also, I think. So I think it's an open question, yeah, but very good. Yeah.
And what do you think about any control repertoires that would be of, I don't know which origin, either simulated or that could be used to train the data and to maybe make those machine learning more efficient?
So control, you mean healthy repertoire, like ground, yeah. Yeah.
Yes, well, highly diverse, let's say all the possible rearrangement that one could identify.
So I think that both the simulation frameworks from the Balchak lab, like Igor and stuff like that, and our immune sim are generating quite good diversity repertoires. I would say at least representing healthy repertoires to a certain degree quite well. With that, I would have to say that we have never tried to distinguish Igor or immune sim generated repertoires using machine learning from healthy experimental repertoires. I think that one should try also. Yeah. I don't hear you.
Sorry.
Yeah.
Yes, I was saying that if there is no additional question, I can't see any question from the chat. Thank you, Victor, for your presentation, exhaustive presentation on the different tools. And now it's my turn.